Dear friends and dear colleagues, good morning or perhaps good afternoon, and above all, welcome to this third module on brain. Today and the following 2 days, we will have very interesting topics, and above all, we will have fantastic speakers. But I leave immediately the floor to the chairs of this module that are Professor Linda De Vries and Professor Luca Ramenchi. Please.
Linda, I'm going. Good afternoon to everybody, wherever you like in the world. I'm very happy to carry on this initiative about the brain on our webinars from WEMPS and Hippocrates Foundation together. We're very fond of this initiative, and we have the pleasure to introduce a big man, a big researcher, a very good friend. So we have a couple of possibilities to introduce very good people, a very polite one, a very correct one. So Pierre has been really a big help for the research in the field of perinatal brain lesions and understanding the development of the brain. He's a child neurologist in Paris. Is the head of a huge and very important research institution like INSERM, Institute of Health and Medical Research, about papers. Very difficult to name all the papers. I would like to name why he's here, because for instance, he's got more than 600 citations on a paper appeared less than 5 years ago on Nature about the role of the inflammation in the perinatal brain. So this is the polite way to introduce Professor Pierre Gressin. The second one is a very good friend, and he always been very inspiring when we sat together, when we speak about science and life. So it is a pleasure. I still remember the first question I produced when I met him. We were playing football at the European Neonatal Brain Club, and we were discussing about the role, I was asking mainly his idea about the role of astrocytes in the pathogenesis of the white matter. So he is in his field, and we're very happy, honored, and we hope to give the best warm and the best welcome to Pierre.
Thank you very much, Luca, for this very kind and sweet introduction. And I would like first to thank the organizers for inviting me, and in particular, Luca and Linda for the very kind invitation. And to share my screen. Is it working well? Luca, can you see my screen?
Yes.
Yeah.
Perfect.
Okay, thank you very much. So the topic I was given is about why is the white matter vulnerable, and I will of course mostly focus on preterm infants, but probably some of the concepts I'm going to bring up might also be with some small changes, might also be relevant for some cases of term infants. Just as to put in context, we've moved from like in the almost 40 years ago from these big cystic lesions called PVL or cystic PVL towards a more diffuse, of course, much less severe white matter abnormalities in preterm infants, especially the very extremely preterm infants. And that was kind of very nicely summarized or reviewed by Joe Volpe some years ago in Lancet Neurology. We see two types of lesions in the preterm infants, especially talking about the white matter. One is kind of a diffuse abnormal signal on some diffusion weight imaging, but overimposed in some of the infants, we also see some very small foci, which might be like the tip of the iceberg of the formerly existing PVL. It might be the very small version or moderate version of the formerly PVA. And this is how it looks like, as we all know. Diffusion wide imaging, you can see that the white is too white to be totally honest. And then sometimes overimposed, you can see some very small foci, which are probably necrotic. This has been shown more recently that if you want to see these kind of necrotic lesions or punctate lesions, you have to do very early MRI. If you wait until the babies that reach term age, very often these punctate lesions have disappeared, probably because they've been kind of resolved. So we have two kind of types of lesion which can be combined and superimposed. One is probably diffuse and probably more maldevelopment, as we will see, and this punctate lesion is probably more destructive necrotic. So we might have two different sets of mechanisms occurring at the same time. We also learned from, especially from epidemiological data, but also from animal studies. So it's probably a combination of different factors which lead to this abnormal white matter. It can be factors before birth, factors around birth, like hypoxia ischemia, for example. Probably also some factors after birth in the NICU, because of course neonatologists have to take care of these babies, very high-risk infants, and of course everything we do to this neonates might also have an impact on the developing brain. But what have emerged, and it will be like a little bit my bias. Of course, there are two main hypotheses. I don't think they are mutually exclusive. I think they very often be combined. One being hypoxia ischemia, and the other one being the inflammation. And we have learned, especially from the Olaf Daman and Anne Leviton's work, that intrauterine infection is probably a risk factor for both preterm birth, but also for abnormal white matter development through the so-called fetal inflammatory response. Because inflammation at the level of placenta and the membranes around the fetus is kind of very well tolerated for one good reason. The fetus has half of his genome or her genome coming from the father. And if the immune system was normal, it would be rejected. There would be no human population on the earth. So we need to shut down, I mean, Mother Nature needs to shut down the immune system around the fetus. So any bug which gets into the placenta will have a potential to develop, even if it's not very aggressive compared to a normal human being. And so this local infection will lead to local inflammation into the fetus. And we know that this inflammation not only will induce neuroinflammation, we'll come back later, but also probably induce this preterm delivery. And if you put inflammation in a broader context, I just mentioned on the left part, the amniotic infection, placental infection leads to fetal inflammation and neuroinflammation, but also the other reasons why the newborn during birth or after birth, the preterm infant can be also exposed to systemic inflammation. Of course, hypoxia and hypoxic ischemic events during delivery we know it leads to systemic inflammation, mechanical ventilation as well, sepsis, of course, and later on, neck can also lead to massive inflammation. And this has been confirmed, but if you measure, for example, inflammatory cytokines in the blood or cord blood, very often there is an increase of the level of this inflammation. And then the hypothesis we and others made that this systemic inflammation will talk to the brain, and one of the key cells in the brain which will see the inflammation in the periphery is the microglia. Microglia is the macrophage of the brain. So it's an innate immune cell, but it's very responsive to inflammation. And the hypothesis that this activation microglia, and we'll come back to this, of course, will lead to brain maldevelopment. And why microglia is so important for the white matter? Microglia colonize the brain during embryonic and fetal development. Most of the microglia progenitors are coming from the yolk sac, so very early on, and then they go in the bloodstream, and then they go into the developing brain. And there are different ways for microglia to get into the brain. They can go through meninges, they can get out of the vessels, which are kind of developing radially in the developing cortex, but also many of them go through choroid plexus and they get out, and then they go in the ventricle and they swim, and then they go out through the ventricular zone, and then they migrate to colonize the whole parenchyma. But unfortunately, for the preterm infants, when they're born preterm, you can see here with immunohistochemistry, most of the microglia, when they're born preterm, are localized in the white matter. A few weeks later, they will be dispersed throughout the whole neopallium, but when you're born preterm between 24 and 28 weeks, most of them are still located in the white matter. And so we and others think that it's because of the location that the white matter might be extremely vulnerable in preterm infants. And indeed, if you look at the stage of activation of microglia, if you compare on the left side a fetal brain of a control, of course, controls are not a true control because otherwise it wouldn't be dead, but it's kind of a fetus who was aborted for non-brain reason, and the brain, at least on a pathological level, didn't look abnormal. You can see the white matter looks totally normal. You can see here in panel J that the microglia is ramified, low density, which is you expect in a developing brain. In contrast, if you look in the preterm infant of the same age, you can see first the white matter is not totally normal. You wouldn't see much on the T1, T2 because it's too microscopic, but it might explain partly why diffuse weight imaging is abnormal. But most importantly, if you look in panel H, you see many more microglia, and also they are like rounded in terms of shape, called amoeboid, and it's a sign of activation. It's kind of a pro-inflammatory activation. So we suspect that this microglia will produce cytokines, chemokines, ROS, reactive oxygen species, which might be harmful for the neighboring cells. So we have a lot of microglia which are still migrating, but they're kind of localized in the white matter when they are born preterm. And on top of this, because for many reasons, they get activated and probably become toxic from the environment. It's a very complex slide, but it's just to remind you that the orange is a term birth, but if you move a bit to the left, it would be preterm. You can see that the brain, but also many other organs, including the immune system, are highly developing. So it's kind of a moving target. And it will be kind of a important theme in my talk. Time is very important. So when you're born at one given time, you have different events ongoing, not only in the brain, but also in the rest of the body. And we have learned now that the brain is not isolated from the rest of the body. It's still a learning process, but we start to understand all the different organs, including the immune system, are talking to the developing brain and vice versa. And this whole chemistry is very important. So timing is very important because if you're born here or born there, you have different events ongoing at different levels of maturity in the different organs of the body. If you focus more on the brain, this is a preterm delivery here in orange, or a very preterm, extremely preterm delivery. You can see there's a lot of things ongoing, affecting neurons, but more looking today to white matter. In terms of white matter, you can see the glial cells, which are highly important in the white matter, rapidly proliferating and maturing. In particular, the oligodendrocyte lineage is in a completely developing level when you're born preterm. You know, oligodendrocytes, mature oligodendrocytes are very important because they myelinate the axons, and it's a very key factor to enhance or to improve or to upscale the way neurons can talk to each other. And of course, it's a major issue in brain evolution, myelination, and so it makes connectivity 20 times faster than without myelin. So myelin is a very important thing during brain development. And so oligodendrocytes are the key cells allowing this process. And we've learned, especially from the Joe Volpe's group in the last 25 years, that these cells go through different steps. They go from a progenitor on the left, and then they go to intermediate immature cells. And finally, they will develop into a fully mature oligodendrocyte, which is the only cell in the whole lineage which can produce myelin. And we have learned that these cells intermediate, which are the most prominent cells of this lineage in a preterm infant, are highly sensitive to all kinds of things like inflammation, oxidative stress, excitotoxicity, etc. So these cells are very sensitive and just on one side immature, but also very sensitive to environmental factors. When people studied cystic PVL, they were able to show that these intermediate cells, due to the high stress they were exposed to, like hypoxia, inflammation, excitotoxic insults, were dying. So there was a loss of these cells leading to immature or lack of myelination. More recently, when we and others look back at the new generation of preterm infants, like the last 20 years, we will realize that in fact these cells don't die anymore, probably because the intensive care therapies have been largely improved and there were much less aggressive treatments, and so the oligodendrocytes are exposed to less severe environmental factors. And indeed, when we look at the numbers, they don't die anymore, at least in most of the cases. However, people have shown, we and others, that in fact, even if they don't die, they get stuck in maturation. At least they get delayed, at least for several weeks, even months. So the cells are still present, so it's no longer an issue of cell death, but still they don't manage to fully complete the program of development. So despite the fact the cells are still there, they don't reach in the right time the fully mature stage, and so they don't produce myelin at the right time. If you look later on, probably they will manage to do myelination, but we have learned from basic science that if you do the right thing, but at the wrong time, the final function can be largely impaired. So timing, again, is very important. So even if you do it, but too late, it might not be the same thing. Is there a link between the activation of microglia I showed you before, which is observed in humans, and the lack of, or the blockade or the delay of maturation of the oligodendrocytes and delay of myelination? Of course, in humans, it's impossible to test this. We can just observe associations, but we can use animal models. And just to make a short or long story, if you delete by 50%, shown here in the right, the amount of activated microglia using gadolinium, just a way to kill microglia. You see here a significant reduction of the activated microglia. You can see here on the right that you can restore an almost complete myelination in this mouse model. So strongly suggesting that indeed activation microglia, because at the right place, at the wrong place at the wrong time, might have a strong effect on the blockade and maturation of the oligodendrocyte cells, leading to delayed myelination. So again, it's a timing issue. It's because of the wrong time, the baby is born preterm, the microglia is the wrong place, they get activated, and they will influence negatively the maturation of the oligodendrocytes. Coming back to Lucas' question, the first time we met, are the astrocytes important? And in fact, yes, we were able to show that microglia here impact the maturation of the oligodendrocytes, but they require a certain cell type, which is the astrocytes. It's not a direct effect. In fact, the microglia will talk to the astrocytes, and the astrocytes will produce toxic factors like prostaglandins, which will impair the maturation of oligodendrocytes. So it's like a ménage à trois. You need more than one cell type to influence the maturation of the brain. It's really a complex chemistry, but again, it's the wrong time, the wrong place, and the wrong interaction between microglia and astrocytes, which will lead to the delay of maturation of the oligodendrocytes. With more recent techniques, like it's a bit, I take single cell RNA sequencing, we and others have been able to show that in fact microglia, like probably most of the cell types of the body, including brain cells, come in different flavors. And so it's a complex slide, but just to tell you that there are at least 11 subtypes of microglia. Interestingly enough, when we look here, these are the controls, and this is here, you can see that some of the cells are disappearing, and some of the cells are enriched in a mouse model of preterm delivery, to say in a large way. And we've been able to show that, in fact, we have two things. Not only microglia becomes activated and will produce toxic factors, as I mentioned before, for the oligodendrocytes lineage, but also we have learned that because there are so many subtypes, some of them are also useful normally in normal conditions to promote the differentiation, maturation of oligodendrocytes. And these cells, microglia cells, which are important and positive for oligodendrocyte maturation, in fact, disappear when you have inflammation. So it's kind of a double hit. Not only the remaining microglia becomes toxic, but also the good guys are disappearing. So it's kind of a, again, wrong place, wrong time. And when microglia gets exposed to inflammation, they do different toxic things for oligodendrocytes. Also, something we have learned from, again, from animal studies is that microglia, if they are exposed to neurons which are activated, they have tendency to take care of the neurons and especially of the synapses. They do synaptic pruning, which is important for the whole network. But if neurons are silent, they kind of get disrupted from their normal function, and then they go to myelin and remove the myelin. So again, it's another way for microglia to be toxic. And we know that in preterm infants, very often they are sedated. They have to be anesthetized for different intervention. And all these drugs, what they will do is to shut down the neurons activity. And of course, if you, and as just mentioned, if you block neural activity, microglia here in yellow will forget to deal with neurons and go again to the white matter and strip the myelin from the axon. So it's another way, and it's linked to the what we have to do in the NICU, blocking the neural activity to do interventions in these preterm infants. So it's another reason why these microglia can become toxic for white matter. One important, it's a very complex slide, it's just to remind me to tell you about the concept. Oligodendrocytes, when they develop, of course, they want to produce myelin and they're exposed to toxic factors coming from microglia, from astrocytes, from other cell types probably, but they're not totally passive cells. And in fact, what they do when they're exposed to inflammation, they start to produce inflammatory factors as well, enhancing the vicious circles. So they will stimulate even more microglia to become inflamed. So instead of trying to block the inflammation, they kind of become crazy and enhance the vicious circle inflammation. So again, because they're probably mature, they're not supposed to be exposed to inflammation at this stage of development, and so they will contribute themselves to their own blockade of maturation. It's kind of vicious circle. So it looks like it's a very complex system. You have different cell types interacting, and it gets worse and worse when you've started and initiated this vicious circle. So myelination, of course, is one very important thing for white matter development, but also, of course, the white matter volume is also determined by the numbers of connections linking different brain regions. So connectivity is also a very important factor determining the microstructure and volume of the white matter. And this one example is a study from the Edwards group at King's showing that, in fact, in preterm infants, if you compare to term infants at the same age, if you look at connectivity, everything which is in red is kind of a part of the brain which have a reduced connectivity compared to term infants. So globally, the brain is much less connected. If you look which are the white matter tracts which are mostly affected, it's a lot of tracts coming from the thalamus, thalamocortical connectivity, or cortical thalamic connectivity, because of course on these images you cannot determine in which direction the connectivity is organized. But for sure, there is a strong reduction of many tracts in the white matter. And we have learned from basic science, again, that in fact, the axons coming from the thalamus, which kind of receive all the senses from the periphery, so visual, more sensitive, everything. The way you interact with the environment is going through the thalamus, and then from the thalamus goes to the cortex. But during development, you can see here, it's a coronal section. You have here the ventricle on the left, on the white part, is the ventricular zone. Is the future white matter here. and in blue you have the cortex. But you have in middle, during brain development, you have a huge purple region which is called the subplate. And all the all the axons coming from thalami at some point get stuck here in this subplate. It's even main synapses in the subplate neuron. And then after a few weeks, they will stop to interact with subplate neurons and go to the cortex to do the final innervation. At the end of the day, these subplate neurons will disappear. So it's a transient layer, but it seems to be extremely important for connecting the thalamus to the cortex and making the right connection. And we have shown that, in fact, the behavior of microglia in the white matter and in the subplate, at least the deep part of the subplate, is exactly the same. So what everything I've shown you so far in the white matter with microglia also seems to occur in the deep part of the subplate. So suggesting that maybe also microglia might have a negative impact on the way axons coming from the thalamus will be impaired in the connection, in the final connectivity with the cortex. So it's at this level an hypothesis, but it might explain partly why we see a very significant disconnection between thalamus and cortex in former preterm infants, further contributing to the abnormal white matter we see in these preterm infants. And yes, and again, coming from mouse studies, which of course do not always translate completely in humans, but people have shown microglia is important for axonal growth, but also for axonal pruning. We know that we have too many axons to start with, and some of the axons have to be removed, but maybe if microglia is overactivated, maybe the axonal pruning, especially the subplate, might be increased in a pathological way, leading to reduced connectivity and abnormal white matter. Again, this is just hypothesis because it's very complex to prove or even support in human studies. As I mentioned briefly before, the brain is not isolated from the rest of the body, and we are learning more and more. And one key thing might be the microbiota. And there are different places where there is a microbiota, but the most studied one in the GI tract. And we know that bacteria, viruses, fungi, parasites, yeast, etc. And in fact, it's a huge number. If you look at the number of genes, our gut contains 100 times more genes coming from non-human origins compared to our own genomes. It's a huge amount of potential information and proteins. And we know that these bacteria and other bugs in the GI tract can produce all kinds of things which can influence locally the GI nervous system. And just a parenthesis, we have more neurons in our GI tract than our cerebellum. So it's a third brain, basically. We have the first brain, which is a frontal brain, the second brain, cerebellum, and the third brain in our gut. And so they produce all kinds of stuff which can influence this third brain, but also some of these factors are diffusible and can go through the bloodstream up to the brain. It can cross the blood-brain barrier. And also we have a vagal nerve, which is a highway between the GI tract and our brain in both directions. So there are many ways that the bacteria, mostly studied of the gut, can talk to the brain and vice versa. And just as a joke, Hippocrates was not a stupid guy, said many centuries ago that all diseases begin in the gut, and I think it was probably partly right, or largely right. And we've learned also that depending on the way you're born, if you're by C-section or by normal vaginal route, your gut microbiota will be very different, because in fact, also there is a debate whether or not there's already a gut microbiome before birth. Most of the colonization occurs during birth, and you get your microbiota from the vagina and from the skin from the mother. If you're born by C-section, it will be mostly bacteria from the surgical room, which is very different for sure. Yeah, you can see. And indeed, if you compare the microbiota of preterm infant versus term, and the plus means that you have an infection, a chorioamnionitis. You can see that there are some common bacteria called phylae, but also very different ones. And so for sure, preterm infants have a very different microbiota for weeks, even months, compared to term infants. And we believe that maybe this different microbiota can also influence the developing brain, including the white matter. In human preterm infants, we have just association, but we can try to manipulate the system in animal models. It's a bit complex slide, but we have in fact 4 groups in this study, and 2 factors. One is vaginal delivery versus C-section, and the other one is control conditions, PBS, and the other one is reproducing the systemic inflammation. So, and the most important thing is when you combine systemic inflammation, IL-1 beta, with C-section, what is the impact? And we can look at the variety or the diversity of the microbiota in these mice, newborn mice, and there are different ways to look at diversity. But if you just focus on the right panel, you can see compared to the control in, it's a purple, I'm not good with colors. But if you look at the red ones, which is a combination of C-section and inflammation, you can see a very massive decrease of diversity. So the two together is the 2 factors separated, which have an impact, but the 2 together make things much worse. And this, if you look at myelination, looking at different markers of myelin, whatever the names, we can see again the red ones compared to the controls in blue are the worst case scenario. So of course, it's just again a mouse model, but if you combine this data with what has been observed in preterm humans, one might argue that maybe the combination of C-section impacting the microbiota plus inflammation might be the most deleterious situation, at least for myelination with the white matter. Again, the brain is not isolated. I've been focusing a lot of microglia, so the brain macrophage, but there's also other immune cells in the periphery. And it's important to tell that if you do a model of systemic inflammation, the blood-brain barrier will open, and things like neutrophils, monocytes, and macrophages coming from the periphery will get into the brain. So again, there is a crosstalk between the periphery and the brain, and most importantly, they will get into the white matter, and so we believe that these cells might also have a negative impact on the developing white matter. So, and very importantly, these cells compared to a term infant or even an adult are very mature, and so they probably respond differently. And again, timing is very important, meaning that because these cells are mature, they might respond abnormally to the white matter compared to term infants or a child. Yeah, and then this is shown here on the light blue. And if you look at the immune system, and again, preterm delivery is here, you can see that there are a lot of things ongoing, a lot of maturation of these immune cells. And we believe that because they're mature and there are emerging data in humans as well, supporting the idea that these immature immune cells might have a very negative impact on the developing white matter in preterm infants. And so we think that it's even more complicated. I mentioned interaction between astrocytes, microglia, and oligodendrocytes, but now we have also immune cells coming from the periphery. So it's like, it's not a ménage à trois, it's a ménage à four, and probably even more, probably endothelial cells I didn't talk about. So the manipulative interaction between many immature cells which react in the wrong way because, again, timing is everything in these preterm infants. So to summarize or to conclude, we believe that, and we and others believe that pre- and perinatal inflammation will induce neuroinflammation, which is probably a very important factor in preterm infants leading to abnormal white matter. Yeah, we understood that I'm microglia-centric or focused or biased. We think they are very important, but as I mentioned before, it's not by itself only. They interact with other cell types like astrocytes, and also just finished by immune cells from the periphery, which are in a mature stage, which I explain why they react abnormally. And also very importantly, the oligodendrocyte precursors at a stage which is very mature and they respond very abnormally. They're very sensitive to toxic factors, but also they become themselves pro-inflammatory. And so we probably, the combination of all these lead to delay of the maturation, delay of myelination. And again, we have to think beyond the brain itself, factors coming from the mother, because I didn't mention it, but the microbiome of the mother and the vaginal level and skin level is also influenced by different material conditions. So mother is also influencing the fetus and the preterm infant. And then I mentioned microbiome is also very important. And something I forgot to mention, if you give antibiotics to a preterm infant, of course, this will have a negative impact on microbiome. So again, we tend to have a vicious circle. And so altogether, it's a multifactorial things. We can identify some key cells, oligodendrocytes, microglia, but they're not alone. They're part of big thing, and the brain is not alone. Again, it's part of the body and also link, a very strong link with the mother. And with that, I will finish here, and thank you very much for attention. So,
perfect timing, Pierre. Thank you so much for your outstanding review and lecture on this subject that is so important for neonatal medicine and for child neurology discipline as well. So, Linda, you're more than welcome, of course. We had one question only. Before that time, you can have some sort of, how to say, suggestion triggering for more questions from the high number of people following. We had more than 370 people following your outstanding talk. Pierre, thank you so much also for being on time. Right. Now let's read the single question that is there. Is regarding it's coming from Elena Lutz. Good afternoon. Regarding sex-specific vulnerability, are there glial cells more relevant to the other?
Very good question. I didn't mention anything about sex, but it's a very important element. It's clear from clinical data that there is a sex bias, and so boys seems to be more affected. Whatever you look at, they're always more affected than girls. When you look at animal models, which is where you can really look at the cell types, and when others have started to look at that, we did not find one key cell which would be sex dependent. It seems more like it's a domino effect. You have at every step you look at, all the cell types I mentioned, there's always a slightly more effect in males versus females, but it's never massive. So we think it's a combination of A plus B plus C plus D, etc., which leads at the end of the day that the phenotype is very different. But we were not able, and other people were not able either, to identify, let's say, microglia or astrocytes or whatever, or even immune cells at periphery, which would be totally sex dependent. It's always a small effect. Almost every step you look at, you see a small effect. But so we believe it's a combination, which is in a way makes sense, because probably all the cells are exposed to the sex hormones, if you believe that sex hormones are important in the sex effect. On the other way around, it's a bit a shame, because in terms of neuroprotection, it would be much easier to have one single cell type to target. It doesn't seem to be the case. It seems to be a global effect, which is a combination of multiple steps.
Thank you. There is quite a few questions. This one is about, very hard to pronounce, sounds to me, Russian surname. What do you think about stem cells, speaking about mesenchymal stem cells in therapy?
Okay, interesting question. there is a lot of work ongoing on the potential use of mesenchymal stem cells, which can be from different origins. We have a European consortium, including Utrecht and other groups, really looking in a kind of detailed and systematic way in different animal models of brain lesions, mimicking preterm infants' brain damage, trying to understand what are the brain damage which could be prevented by MSCs, mesenchymal stem cells, and also, very importantly, is it a prevention or could it be a repair? I would say work in progress, but based on the fact that these MSCs can release trophic factors, anti-inflammatory factors, plasticity factors, immunomodulatory factors, at least it looks like a very good potential strategy. But I would say that most of the work has been done so far on the stroke and term infant brain damage or IVH, but not so far as I know, nothing really on the white matter. So I think it's work in progress, but we believe, because we have a project on that, we believe it should be a good strategy, but I would say work in progress.
Thank you. Again, another question, just to give you an idea to whom you're speaking, a question from India. Dr. Varma, does anti-inflammatory agents have any role in the white matter injury? I think this is an assist for Professor Pierre Gresson.
Yeah, it's a very good question. Initially, when we started, I mean, we and others, again, that 20 years ago, we started to work on neuroinflammation. We said, okay, blocking inflammation will be the key thing and should work. Unfortunately, as often, things are more complicated than we expected. Let's say one example, and it's probably true for many immune cells. If you look at microglia, we have learned that they come in different flavors. And I briefly alluded that some of microglia positive, supporting oligonucleotide maturation. So if you just shut down microglia, you might have beneficial effects because you will reduce the amount of toxic factors released by them, but you will also block potentially positive effects of some subsets of cells. So we've moved, when the whole field has moved from just blocking, like with steroids, for example, just blocking microglia. Steroids are very efficient. If you put steroids, activation is almost close to zero, but it doesn't mean you improve the phenotype. So we've moved from this total blockade of microglia activation or immune cells towards a modulation of the activity, trying to switch them from a toxic inflammatory phenotype towards a pro-developmental phenotype, which is of course much more complicated. But we have some clues. For example, we have learned, mostly from immunologists, looking at macrophages, when they are inflammatory, they have a tendency to be using glycolysis for energy. While when they become pro-repair or for anti-inflammatories, they use lipids as a metabolic substrate. So the idea now is to try to play with the metabolism to try to change the phenotype rather than just blocking them. Because if you block them, you might have an early positive effect, but at the end of the day, you either neutral or even toxic because you block the repair capacities of these cells. So it's a complicated thing, yeah. But it's when I say we, it's the whole committee. We are starting to understand the different phenotypes of immune cells and also the timing. Again, the timing is very important. There is an acute phase and a delayed phase. You might target different things. So again, timing is a big issue.
To honor all the people that are making questions, there is from Silke Stekeda. You already replied to the mesenchymal cells and delivering cells, but can you add something about the potentials of timing? Because you already mentioned that how important is timing. So in case you figure out when it will be the time.
So with mesenchymal stem cells, that, I mean, in a simplistic way, that two windows two potential windows for intervention. The first one is during the acute inflammation. So you have the systemic inflammation inducing acute neuroinflammation, and you want to, like the fireman, you want to block the inflammation. So MSCs might do the trick, because at least in vitro and other models, they've been shown to be able to release factors which will dampen the immune system. The difficulty with that in preterm infants is that you would need to administrate the stem cells in an acute phase when the baby is very unstable, very immature, and also you don't know exactly which baby will need treatment, because some of them will do well whatever you do. So there is a kind of a bit of an ethical issue, but you want to treat a baby who doesn't require anything with stem cells, where we don't know exactly what will be the very long-term effects of these cells. On the other hand, if you wait a few days, weeks, you end up doing the pro-repair, or when the brain is trying to adapt to the initial damage or try to catch up. And then again, MSCs and mesenchymal stem cells might be interesting, because they're known also to produce a lot of trophic factors, which might help the brain to recover quicker. And at a stage where you know that the brain is abnormal, so you suspect that the outcome will be impaired, and also you're kind of away from the acute stage, and the baby is more stable and have grown up a little bit already. So that maybe both together might be interesting, maybe a combination of the two things might be interesting, but I would say in a simplistic way would be the two different approaches.
Right. Thank you so much for this reply.
Do you think, Pierre, that for instance, if a child develops a septicemia or a necrotizing enterocolitis, that you would repeat giving the stem cells?
That's a very good, it's very good hypothesis. Yeah, I think so. I think if you can show that in the early stage it's protective, I would do it, especially in the neck, or in both cases. I think the level of inflammation is much higher than the regular preterm infant. So yes, for sure. Yeah, very good. Yeah. Of course, in animal models, it would be extremely difficult to mimic, but then only clinical trials, I think, would be able to address this interesting question. I would say yes.
I'm in doubt what to do. So to read the closest question about this aspect that you already mentioned with Linda intervention, no? And so going in this direction, I'm jumping from a list of questions. Are there differences between brain development in the fetus with infection if they receive antibiotic or not? Victoria Lima from Mexico. So, I don't know, links to this area.
So much, if I understand what's the question, as there has been, and probably you and Linda can comment more on that, a few clinical trials trying to give antibiotics systematically when there was a close to be born preterm. And there was no positive effect on the outcome. It could be because the hypothesis is wrong, that in fact inflammation or the infection doesn't do anything, it's just a proxy for something else. So it's one hypothesis. Second one is because the bugs that they develop in utero are not sensitive to the antibiotics we give, because as I mentioned, they're probably very weak bacteria, but they develop because there is no local immunity. Our third is because it's too late. Once we pick up a suspicion of infection, it's already inflammation, and so antibiotics is too late. Or number 4, I don't know, something else.
It's more than enough, I guess. Close to this area, there is a nice question from Medina Pilar. Is there a way to favor high neuronal activity in the preterm baby as a way to reduce microglial activity?
I think
all people, they wish to work with you, Pierre, I guess.
Yeah, I mean, I kind of induce the question by, I would say, and again, under the control of you and Linda and the other neonatologists in the room, I'm sure I would play with the electrical activity, because then we might be running the risk of seizures and other things. So I was more alluding to this fact because it's based on experimentation. Maybe what might be interesting would be the other around. If a preterm infant doesn't need sedation or strong sedation or anesthesia, maybe to try to think if it's useful, then of course you have to do it. But if it's not useful, maybe think that, as always, I mean, if a baby doesn't require something, maybe not to think that it's safe. Everything we do, if it's not useful, we might think twice about before doing it. I would put it more that way than the other way around, and I wouldn't play with overstimulation, because I think I think we run a big risk.
So you didn't use the word pain or nociception. That is even more vast. Anyway, Eric Ben, you may know him. Great talk. What specific evidence is there to support the maturation arrest hypothesis? If the subventricular ventricular zone response is appropriate, one might expect to see increased OPC. In human autopsy tissue, it is impossible to determine if these cells are truly arrested.
It's true. I mean, of course, yes, in human situation, we have one given time point, one fetus and one age, and another one at different age, or one preterm. So of course, we don't have the complete history. The only thing we can say, we don't see markers of cell death. We see a normal number of the whole population, and we don't see changes in proliferation. So it's indirect evidence. In rodents, we can play more with a model. And in fact, if you do a moderate or mild inflammatory stimulus, you observe the same thing in humans. You see no change in proliferations, no change in cell death. Total number is okay. And you can look at markers and you see that more immature forms and less mature forms. And you see a delayed myelination. So it's in rodent model. On the other hand, if you use a more aggressive model in rodents, where you induce cell death of the oligodendrocyte progenitors, so they die in this case, you see an increased proliferation, which is probably a response of the brain to try to compensate. And interestingly enough, you also see a delay in myelination, but in the mild model, it takes, I would say, 10 weeks to recover. They will myelinate with some delay, but they will catch up at some point. It takes 10 weeks. In the more severe model, they take only 5 weeks to recover. So if you hit hard on the brain, if you don't go beyond the capacities to compensate, they compensate quicker if you do a mild system. So and it might be the same in human beings. Of course, it's very difficult, but because we have a tendency to combine all the patients. But maybe if you're looking at the mild cases to start with, do they recover or do they catch up quicker or in a more delayed manner compared to not the very severe? Of course, the very severe they won't recover, but there might be a U-shaped curve, which you might have missed, because it's very difficult, of course, to determine who is mild, who is moderate, who is severe. Severe is relatively easy because they have early brain damage. But between the mild and the moderate, and to see the outcome, it might be missing something, at least based on animal model. But of course, rodents are different from humans. But yeah, it's a long answer to the question, but it depends on the severity of the insult, at least in the rodents. And we have, at least in the rodents, again, strong evidence there is a delay in maturation, based on the combination of different things we can do.
Pierre, we have more questions. So are you happy to reply to them as well? We have more questions. Are you happy to carry on? We got a few more minutes. No, I can read.
Yeah, sure.
Yeah. Just to help you out. I mean, blah, 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 blah. We mentioned that one. Gerson Theophilus Lazarus. Good morning. Thank you for an extremely informative talk. Do you have any thoughts, data? We're going back on perinatal world. On the impact of maternal intervention, invasive and non-invasive, biofeedback, medication, on preterm brain development, specifically intervention and fetal brain development. So I don't know if there is any thought, just to give you time, to reply to this question. I was very fond thinking the way you delivered this very nice talk, because everything looked pretty simple, simply because you were speaking about it, but it's so complex. And you mentioned no right thing at the wrong time as an issue. And I was very impressed by a talk about, from yours, that prematurity per se may impair the development of the cerebellum. With your high experience, do you think it is a similar issue for white matter or not? In a very simple way to ask a question.
We compared in the same animal models. Most of the work we have done and other people have done so far was on the frontal brain. Very few people have looked at cerebellum, so I think it's a shame. So we've started to look at cerebellum and compare. It's the very same model, and it looks like the cerebellum is more affected in a way. The white matter is more severely affected than the cortex, neocortex. And in fact, why we don't see cell death in the neocortex, we see significant cell death in cerebellum, which is exactly the same insect, but the same brain, basically. So yes, cerebellum seems to be more sensitive, at least again in mice, in rats, than the forebrain. So I think we should put more investment in the cerebellum, which we know now is not just for motor but also for cognition. So it's very important. And so we might have missed. I mean, not completely, because there are some data showing clear effect in cerebellum in correlation with outcome, but we used to, in animal models, to use cerebellum to fix the brain in the block, and I think in many MRI data they use cerebellum to normalize the data. So I think very often cerebellum is lost, which I think we should change our practice.
We may simply follow you.
So yes, cerebellum. So there's a question. I think it's for me it's a completely black box. So I have no opinion, but I think we have to be careful when we try to do anything in utero. I think it's a...
Yeah, there is a balance, for instance, for steroids. Steroids are very good for promoting lungs, but if you use a bit more, perhaps it's more dangerous than anything else. I'm not sure is a question to that. Is an answer to that question. Is the a way to reverse the we're still there. Oligodendrocyte arrest after it happens. Can you promote them to resuscitate the arrest of oligodendrocyte?
So there are a lot of knowledge on the maturation of the oligodendrocyte lineage, not for preterm, but as a basic neuroscience. So we know a lot of pathways which are involved in the, and it's a very complex thing because you have multiple steps, and each step is necessary for the next one. So it's not you cannot jump from A to Z. You have to do from A to B to C, etc. So it's a very complicated story, and so I'm not sure we know enough in the human preterm infant. We know they're probably delayed, but we don't know exactly which is the steps which is really impaired. So before and until we know exactly which step or steps are really impaired, it's going to be difficult to directly intervene on the oligodendrocytes. Having said that, at least in the mouse model, if we target microglia or astrocytes, we can remove the brake, and we can let the oligodendrocytes mature faster. So probably at the level of our knowledge, it might be easier, but maybe 5 years from now, target directly oligodendrocytes, but right now we don't know enough of the human situation to determine which is the steps to be targeted in oligodendrocytes.
Thank you so much. And I know that we have people like Linda that are very, very good to keep things on time, but we've been allowed 5 more minutes. And there is another question that is going to make a bridge, I guess, between the gut, as you said, you know. And Monica Morgus is asking if there is any clue about colostrotherapy make a difference in cerebral immunity. Do you have any data?
I think Joseph is probably much more knowledgeable than me on that. I would say that, to my knowledge, there is no real clinical trial, or at least a controlled study, looking at the effects of pre or probiotics or meconium, whatever, or feces transplantation in preterm infants and looking at the outcome. I know that, at least in France, for example, many units give pre or probiotics systematically, but there is no data to really make sure it's useful or not. But maybe Linda or Joseph or you know more than me on that, but yeah.
No, not really, personally. Anything else?
It's probably an avenue for future research, but I think as always, it has to be done correctly. Otherwise, we can keep giving pre or probiotics forever and never know what is doing, if it's useful or not. It's expensive for sure, but is it good? Maybe.
I'd like to say 2 things. Up to now, I think this has been the most, how to say, I'm looking for the right adjective, not only entertaining, but really, really impressive discussion, very, very focused. So the quality of the questions are probably related to the quality of the speaker. So I think these have been very good questions. So that's the way to say thank you to Pierre and all the people attending the meeting. And can I make a joke, a sort of secret joke with Pierre? Can you allow me?
Sure.
It's always a pleasure to interact with very good, fortunate genotype.
I got it.
Okay.
I see Linda as well.
All the best to all of you. And
thank you, everybody. That's great.
Thank you so much, Pierre. It's always a pleasure. And we had really a lot of people attending up to now, even at this time. So thank you always so much. Now it's the time to introduce the next speaker for tomorrow. And I hope we have all the people such as we had today. So for this very interesting talk, imaging, we go back to our, how to say, first love. And early outcome white matter injury. So we hope to have all you tomorrow with Professor Linda De Vries. And we say thank you to everybody. Linda, bye-bye, thanks. And Corrado, thank you for this initiative again. We think today we had a very nice time all together. Bye-bye, see you tomorrow.
Bye.